Hello children, welcome back to my talks, and here I am with standard 12th biology. Okay, so molecular basis of inheritance we were doing, and we have reached the machinery of the enzymes. Okay children, and this machinery of the enzymes we are going to start today. So today I've like many of y'all told me also, okay, and I thought that okay, let me try as we have been doing in the class, okay, complete explanation, because definitely the questions or during your board exams and even in the NEET, okay, you must be thorough with your text. That is what it says. Okay, so if some of the points are left, normally I tried to teach you in the lecture way, okay, but okay, let me start with this today, and you tell me whether you are enjoying this way or the other way. So the topic is 6.42, that is the machinery and the enzymes. Okay, so what does it say? That in living cells, the E. coli, the process of replication requires a set of catalysts. Yes, the DNA replication we had studied, and the DNA replication or any work in the body, okay, requires enzymes in order to speeden up that particular reaction. Okay, so here it says that it requires a set of enzymes, and the first enzyme, the most important, the main enzyme is the DNA dependent DNA polymerase. Okay, so this is the main enzyme which actually participates or which actually plays a vital role in DNA replication. And besides this, there are other enzymes also. Okay, so what happens DNA, you know, when we talk about the replication, and when both the strands, two strands of DNA are there and they act as template. When they replicate, both of them will act as templates to form another strand, the complementary strand. So that is what it says, and these enzymes normally they are very efficient in catalyzing the polymerization. It means ये वाला जो strand है, ये strand हमारा original strand है, यहाँ वाला strand form होगा. Okay, suppose this is the original strand A, suppose T A G C, and the other strand was, okay, this A. For T it will be what? For A it will be T, for G it will be C, and this G. So these are the two strands after replication. Okay, so now the strand which it will form, this will form one strand and this will form another strand. Got it? So these two both have the capacity to act as template strands. That is what it says. And the enzymes, they play a very important role in getting the polymer, in polymerization, in forming the complementary strand. Okay. So that is what it says, that these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides. Yes, okay, this chain is not just 4. I have shown you in the example. It is very long, and you know that DNA is a long chain, isn't it? So, and here it says that E. coli has only 4.6 into 10 to the power 6 base pairs, okay, while human beings, a human cell, that has got diploid content, that is 6.6 into 10 to the power 9 base pairs, okay, and it completes the process of replication within 38 minutes. That is what it says. तो एवरेज कितना हो जाएगा? The average rate of polymerization will be about 2000 base pairs per second. मतलब एक सेकंड में 2000 base pairs बनाने की कैपेसिटी होनी चाहिए. Okay, and all these polymerases, okay, have to be very fast, but they also have to catalyze. Catalyzation have to take place. The reaction has to be very fast. Okay, and that even with high degree of accuracy. अगर यहाँ पे T है, it requires excuse me, it requires A over here. Okay, here it is A, it requires T actually over here. और by chance T के बदले A हो जाता है. By chance, this is by chance. Okay, that A goes with A, then that will be mutation. That will be a change. Got it? So it has to be very accurately forming the pair of nucleotides. Purine के बदले pyrimidine जो होना है, वो exact होना है. So that is what it says. That with high degree of accuracy also is required. And any mistake which occurs during this replication would result into mutation. I told you. Okay, then replication is very expensive process. Yes, The complete replication means the DNA is a long chain, and अगर replicate होना है तो suppose this I hope you must have understood. Okay. Suppose when the replication has to take place, the whole replication, अगर इतना बड़ा chain है तो ये पूरा का पूरा replication एक साथ नहीं होगा, because it is a very expensive process. That is what it says. Okay, that it is a very expensive process. And finally, the deoxyribonucleoside triphosphate, okay, that is there, deoxyribonucleoside triphosphate, it serves two functions. Okay, this provides energy also for the polymer, for the formation of polymer, that the chain of nucleotides, and it also acts as substrates. You very well know this nucleoside, phosphate, triphosphate, diphosphate, ATPs. Okay, when ATP breaks down to ATP, triphosphate gives ADP, diphosphate, and releases energy. So here also this deoxyribonucleoside phosphate acts as substrate and also provides energy for polymerization reactions. Okay, same as ATP. And the next one, it says that the DNA dependent DNA polymerases, okay, that is required to complete the complete process of replication, okay, and with high degree. Then it says that for long DNA molecules, the two strands cannot be separated in the entire length. Yes, what it says is that suppose this is the complete DNA strand, जैसे मैंने पहले बताया आपको कि ये strand है, तो ये पूरा strand एक साथ replication नहीं होगा. This will not replicate the complete thing altogether. So finally what? It has to be formed in the form of a replication fork. So remember, I took the book so that you don't understand that I'm missing something. So I have done that. Now we come to this replication fork. When we come to this replication fork, we have some other enzymes also. Okay, so we have this DNA ligase. This is another enzyme, okay, which helps in joining ligase to ligate. Okay, now replication fork जो है हमारा, these are the two DNA strands. Now these two DNA strands, as it runs anti-parallel. Okay, one is named as 5-3 and the other goes like 5 up and 3 down, 5 down and 3 up. Okay, and this 5-3 is known as continuous synthesis. So here the synthesis takes place continuously. Okay, and these both act as template DNAs are there. Okay, and the other side where it is 3-5, okay, here it takes place like normally what we do is हम इस तरफ से पढ़ते हैं 3-5. So this is a continuous synthesis while 5-3 is discontinuous synthesis. Got it? 5-3 is discontinuous and 3-5 is continuous synthesis. Okay, and these two strands are newly synthesized. Discontinuous synthesis, now the question must be in your mind that what is discontinuous synthesis? Now discontinuous synthesis, here the strand, the formation, the complementary strand which is formed, okay, that is in sections, fragments you can say, that is formed in fragments, and those fragments are joined with the help of DNA ligase. While here it goes on forming one after another. Here ligase enzyme is not required for the formation of the complementary strand, while in discontinuous synthesis, okay, where it runs from 5-3, there the DNA ligase enzyme is required for the joining of those fragments of nucleotides which is formed, the complementary strand. Okay. So this is one thing. Now here it again talks that it goes off slowly. धीरे धीरे ये दोनों separate होता है और complementary, यानी आप ये बोल सकते हो कि replication as well as the formation of the strand both goes on together. Both goes together. यहाँ से वो separate भी होता रहता है और formation of the two strands also. But one is continuous and other is discontinuous. The most important point is continuous synthesis 3-5 और discontinuous, वो remember करने का है. उसके बाद it says that origin of replication. Okay, there has to be a point from where the replication has to take place. It is not that DNA strand में कहीं से भी replication शुरू हो जाता है. Okay, that we'll see when we do the recombinations and all that. We are going to do quite a lot over here. Okay, in biotechnology it is there, ORI. Okay, so there is a point in the DNA from where actually the replication starts, which is known as ORI. This is known as ORI. Okay, origin of replication. Origin of replication, where actually the replication originates, where it starts from. Okay, and then what it says that in normal cells, in eukaryotes and all, in us, okay, where the cell is well defined, the organs are well defined, cell membranes, okay. There what happens, the replication, you know children, that replication of DNA takes place during cell division. Okay, the cell division which takes place. So here when does it takes place? During the synthetic phase. During the synthetic phase of the cell cycle. Cell cycle में synthetic phase होता है. उस phase में आपका replication of DNA होता है. Replication of DNA has to take place. You know it very well. Okay. So why it has to? Again, just go flashback and let me know. And the cell division and DNA replication, okay, that is coordinated. The cell division and cell replication, यानी सबसे पहले DNA replicate होगा और DNA replicate होने के बाद cell plate formation होगा ताकि divides into two cells, okay, two daughter cells, so that they have equal number of chromosomes. अगर cell plate formation नहीं होता है, सेल ये रेप्लिकेशन हो गया, डीएनए रेप्लिकेशन हो गया और सेल डिवीजन नहीं हुआ तो पॉलीप्लॉइडी विल रिजल्ट. व्हाट विल हैपन इन द सेम सेल, देयर विल बी टू सेट ऑफ क्रोमोसोम्स, टू सेट ऑफ डीएनएस. ओके, तो वहां पे 2n के बदले क्या हो जाएगा, 4n हो जाएगा या 3n हो जाएगा. सो दैट विल हैपन एंड दैट विल अगेन बी वो पॉजिटिव साइड नहीं है, फिर म्यूटेशनल, देयर बी सम डिफॉर्मिटीज, okay, some problems will be shown. So that is what is there. So it's very important that DNA replication and cell division are correlated, they are coordinated. As soon as replication takes place, after that the cell division has to be completed, cell plate formation, it has to divide into two. So these two are coordinated. This is how actually children, the enzymes play a very important role during DNA replication. I hope we have done it. Okay, I wanted to do only this portion, and in the next class I'm going to do transcription and transcription unit. Again a very important topic. Okay, replication folk, now please pay lots of attention about it. Okay, it is asked for 3 marks question to draw, to explain. Okay, so these are the points. I'm going very slow children, only for your benefit, so do benefit from it. Again, as it is, do like it, share it, and if you have not subscribed, subscribe it, and do keep watching my English videos also. Thank you.